very much. And yes, indeed, I'm here to talk about this IMI consortium called eTranSafe. You see the different people that are involved in this, and it does fit, of course, in these new approaches, methodologies that we've been talking about. Considering that we are delayed, I will just fly through the concepts, and I'll be happy to take details during the questions if possible. So you see the title here. This is really what it is about. And if I go next, I will start with this, which is a slide I did a few years ago, but I'm still using it and I love it. This is with the initial statement, you know, when people say, well, drug development, that's not rocket science. I say, yes, you're right. It's not rocket science. It's much beyond rocket science. It takes more people, more years, more time, and more money than to launch a rocket on the moon. And it's more, it's like, you know, 2 to 3 times more than actually launching a rocket on the moon. You see here the details. And the thing is that it's getting more and more. It's always increasing, as I guess you all know, and that is not really sustainable. The thing is that the amount of data that are produced to bring a compound to a drug is absolutely humongous. And it's all geared at bringing that compound to become a drug. Once it's done, it's kind of, you know, all the data generated are slipping in private archives, and that's it. So the big question that we asked in this eTranSafe consortium is, what if we were able to reuse all those data? Because we know, I mean, it has been described several times, that in these kind of cases, 1 + 1 is equal more than 2. And we can certainly extract more knowledge, more understanding of mechanisms of toxicity, of AOPs, of everything you want, by bringing together all those data and analyzing them. So I'm pretty glad that Nicole gave a number of concepts and explanations that will be very resonating here. So it will save me time. I will not need to re-explain them, but you'll see what I mean when we get there. So very briefly, when I will be talking about preclinical here, be animal toxicology data and also in vitro assays, whatever types of in vitro assays. It can be, you know, Ames test. It can be like we've just heard. It can be, you know, other things. It can be off-target things. Okay, so that's all that kind of things, or more complex things like, you know, organ on chip or things like this. So that's preclinical in my mouth for the next minutes. Clinical is human pertaining data, so clinical trials, PSURs, you know, pharmacovigilance, that kind of things. Okay. So I just said there is a huge amount of data which is totally untapped. It's sleeping there. It's, in my view, it's a shame that we don't reuse them. So there is those private data held by the pharmaceutical industry worldwide, but there's also other types of data that are used, but not, you know, not as much as they could. Although we had Nicole, you know, mentioning a few of them. So the thing is that, okay, if we are bringing this all together, we could, in the end, do what is really the heart of the eTranSafe, is to make a very global, thorough assessment on how much the preclinical data are predictive for human safety and how they can be used for human safety. In the late 90s, beginning of 2000, there were a series of papers that tried to do that. It's a Olson and collaborators series of papers, but they were not able to do a very exhaustive analysis because they could use only the few viable data they could put their hands on, and that's a tiny bit of the tip of the iceberg. So that's what we want to do with this eTranSafe consortium. Of course, there are many more aspects into it, but that's really the heart. And here you see the partners of this consortium. You see that they are, like all IMI, you have academic partners, you have small and medium enterprises, and you have big pharma, big and small. I'm pretty pleased that for this one, we managed to attract like American companies like AbbVie, or Japanese companies like Eisai, and this is kind of a new thing in the IMI consortium. Right, so here is a rather busy slide with a list of what we intend to do or what we have started to do. but on the left-hand side, you have the extract of this. So as I said, we want to reuse existing data. We are not generating new data for this consortium. We use historical data and data that are currently generated to bring compounds to the market. The other thing that we want to do is to put in common all those data. So we want to do data sharing, and that by itself between usually competing companies, raising a number of hurdles that we managed to go through, and that was a big step forward. We want to do mechanistic safety. As I explained before, you know, if you bring all those data together, we should be able to get a much better comprehension of what's going on in terms of toxicity. So AOPs and, you know, all the like. Then what is the heart? As I said, you know, do this translational safety from animal to human data, but also the reverse, from human data to animal data. You know, what did we miss or what is not matching, basically. With all those data, we also want to do predictive safety, right? If we are able to bring enough chemical structures, enough associated data, we should be able to build predictive in silico algorithms. All of that should have a relatively big impact on 3Rs. And last, but pretty important as well, we want to propose new guidelines for data sharing based on the previous consortium called ETX and this one, to propose to the world scientific community, whenever you have private data that legal departments or, you know, IP protections are getting in the way, how to actually be able to share those data, even if it's difficult. In previous talks, I called that, you know, sharing the unshareable, but that's pretty much what it is. So where are those data? So preclinical studies, it's private databases, there's no question. Clinical trials, if they succeed, they eventually end up in public databases, just like pharmacovigilance. But the failed clinical trials, the ones that are most interesting to us, particularly if they fail because of tox, these are not publicly available. And they're also in those private databases. And the difference of size here in decibels just exemplify the amount of data on each side, what is publicly available and what is privately held. Very quickly, sorry. The ETX database, which was the, or the ETX consortium, which was before the e-TRANSAFE, was really geared at preclinical only. And at the end of the project, we thought that we're missing a big part, which is obviously the human side. So that's e-TRANSAFE. And the ETX data, which is sizable, will be reused, is reused in e-TRANSAFE, but not only. We are trying to tap in all possible available data or databases that are around, including, you know, the health authorities like FDA, EMA, and PMDA. I will fly above that, but that's basically the new structures that we are implementing for this e-TRANSAFE. We call that the ETX Hub. It's a much more complex system that we had in ETX, and also much more current technology. The ETX, you know, that we started like 10 years ago, the technology that we used at the time is completely obsolete now. So we're starting on a completely new basis with this e-TRANSAFE, which is also bringing me to mention that the sustainability of the system is extremely important, because we don't want to have a dead system after 5 years of collaboration, of, you know, the IMI life phase. It needs to, you know, the life needs to carry on after those 5 years. So we have also big projects for sustainability, but I will not say more than that for the time being. Okay, so what kind of hurdles, and just to name a few, we are facing? Preclinically, we use a certain set of experiments, we use a certain set of endpoints, we use a certain language, which is completely different from what the clinical science is using in terms of setting of the trials, of monitoring the safety, the way to assess the endpoints, and of course the vocabulary is completely different. And overall, what we're trying to do here is to match histopathology, so organs that are sliced, diced, stained, observed, described by pathologists, to symptoms, because usually we don't use to slice and dice humans at the end of a study. We keep them alive, usually, and therefore we're not comparing like for like, and that by itself is bringing a huge complication into this project. Now, as I mentioned before, there have been some attempts to compare preclinical to clinical, but there are a lot of hurdles, and you can just compare, at a high level. I'll skip that. I'll go straight to the next one to exemplify. And this is a paper from Peter Greaves that follows, the one from Olson and collaborators. So here, for example, Peter Greaves made this assessment where, for liver, for example, he was able to assess that about 50% of the studies, all the studies preclinically together, are able to predict what will happen in human by about 50%. Okay, so when I say that, usually people are telling me, well, that's really poor because that's not better than tossing a coin. And then I really raise my voice against that, because no, it's not like tossing a coin. We're not trying to predict, black or white, 1 or 2, or 0 or 1. It's more like a multiple different parameters, and we need to think about rolling like a multifaceted dice, like I put a picture here. And in half of the rolls, you are right. So it's much, much better than tossing a coin. And honestly, if I had a system, such a system like that, was able to predict 1 time out of 2 the result of the lottery, I would play, no doubt. So fine, but for drug development, as we saw, it's not good enough, and we need to improve that, unfortunately. Oh, well, it's part of our job, right? So already mentioned by Nicole, the ontologies, the mapping of the terms. This is what was done in ETX, where the SEND terminology, which is now a global format for preclinical safety, was not existing at the time. So we had to create our own ontologies. And of course, we are discussing with the CDISC people that are in charge of the SEND formats, and we are exchanging information and things. But just to exemplify, if you look at the EMBL database, you will see more than 1,000 synonyms for paracetamol. So you cannot do data mining with this. You need one preferred term, right? And acetaminophen for the Americans. But you need one preferred terms that automatically in the background, if you enter the preferred term, either acetaminophen or paracetamol, that all these synonyms and all the data attached to those synonyms will automatically come up. So there is a fair amount of work there to be done. And just to show how creative we can be, in the more than 8,000 studies that we had in ETX, we found 123 ways to say male or female, which is not bad, huh? And we managed to reduce that to 3, which is male, female, or both. Just to show, the complexity of that. So think about histopathology or things like this. It's of course huge, and you can see, the number of synonyms and how many preferred terms we were able to reduce, which is still a lot, but that makes sense because there is a lot of different talks that can happen. So now we use the SEND format. We work with those guys, no problem, but SEND does not cover everything. And as also mentioned by Nicole, the SEND format is only covering the data, but not the interpretation of the experts that have written those reports. So we have created a SEND-like domain just to put and place and extract the expert interpretation of those data. We don't want to reinterpret studies. They have been interpreted by people who knew what they were talking about, but we want to use this interpretation particularly for its observation link to the compound, right? So that's one kind of things. And to do that, and then I can skip because text mining was already explained, but that's precisely what we do as well. We've been investing a fair amount in text mining through the process that Nicole explained to retrieve the actual findings and interpretation of the findings, either in the abstract or in the conclusion or in this discussion. And this is a big endeavor, but it's necessary. And then you have to do the translation, what we call internally the Rosetta Stone project within the TransSafe project, which is how to match the terms we use preclinically with the term we use clinically. And I just use a relatively simple one, which is hepatic steatosis in human. And that's, the different ways we describe that preclinically, the different clinically, and then the diagnosis which is posed. hepatic steatosis. Here on this we have liver, we have lipidic evacuation as preferred terms, because there are plenty of other terms behind, but these are the preferred terms. And then we can map relatively easily liver with the liver structure and lipidic evacuation with fatty degeneration. And then this SNOMED clinical trial ontology is able to make the link between that stone so that we can do this analysis. Just to give an idea of the concept, but there's a huge amount of work behind. And actually, this is a large part of what we do in e-TRANSAFE. Now, also for data mining and data analysis, you could bring back the data after search. You know, we are developing search tools that can bring endless Excel sheets. And if you want to analyze that with your eyes, you very, very quickly become crazy. So we need those visualization ways to look at or zoom in, you know, like overlook high level, like helicopter view, and then spot what looks like interesting and then drill down and zoom in rather than scrolling through Excel sheets. So plenty of different ways. It's Optibrium, our partners, that is doing that for us, like this Corona visualization where you can zoom in and zoom out. You can actually visualize the whole data from different databases like this, and then drill down. Classical histograms, more 3D things like a chemical space, and we'll talk a little bit more about that, or more fancy stuff like this. All of that needed and quite complex because we have very heterogeneous data. From all those data, we also want, as I mentioned, you know, to build predictive algorithms. And one of our partners from Barcelona, University of Pompeu Fabra, developed this FLAME framework where all you need is a data set. So even people that are completely IT illiterate like me, if I have a data set, I can actually build a predictive algorithm. I just need to click, you know, build model, and that's it. And you see a few that have been built here. And you can then, out of that, you have a number of characterization, you know, the true positive, you know, the sensitivity, specificity, etc. The data set that you've used in terms of positives and negatives, different ways to look at that, and also this chemical space where by itself is already a bit predictive. Here in blue, you have the compounds that are negative for the endpoint of interest, and the red one that are positive for the endpoint of interest. And if you put your own compounds in there, you will see already if they fit more like in the red part or in the blue part. But of course, the predictive algorithm will tell you that much better with a certain level of precisions. Another thing that I wanted to mention here is that it is an idea that was brought by Bayer, by Thomas Steger-Hartmann, who is the co-lead on this thing, on this consortium here. If we were to bring all the control data from all those animals preclinically, there is no issue of confidentiality because there's no compounds, there's nothing, that's just the data. Then we could have a very, very large set of control animals with a very powerful statistical power. And then what we're trying to do here is to think on how we could use this mass of data, since we're always using the same kind of animals, instead of the actual animal group, control group. And if we were managing to move there, we could certainly immediately reduce by 25% the number of animals in our studies, because we could have a virtual control group instead of real. Of course, it will take time and a lot of validations needed, etc. But there are some guidelines that we can base our work on, and that can help. And also, you know, we have Elsevier as a partner who is used to this kind of concept and used to this kind of data manipulation. I mean, manipulation in terms of handling, right? Not in terms of tracking. Basically, this is a work which is ongoing, but it looks good, and the companies are participating to this. So I know I flashed through, but I know we're short on time. So this is the outlook already. The system, the TOX Hub or Knowledge Hub, not definitive name, will be able to access multiple databases at the same time and use one tool, one search tool to actually search in multiple databases with different types of data and different types of formats. thanks to the ontologies and what we call the semantic services. We will need those complex visualization, as I just explained, to search the translational and retrotranslational, but also in terms of safety biomarkers. We believe that by making this in-depth analysis, we should be able to find potential new biomarkers of toxicity. We want to work a lot on the chemical structures and in chemical space, although biologics and new modalities are not excluded from this consortium. Okay, I mentioned that as well, and of course the predictive algorithms. Currently, we have more than 10,000 practical studies in SAND or historical data that have been sanctified. We have off-target, and we start to accumulate, human data to make this translational analysis, which is great. Project is already well advanced of time. We have like a year and 2/3 still remaining, but everything is currently accelerating, which is great. You can follow us, you can reach our website, you can follow us on this blue social thingy. You can ask to receive our newsletter. And with this, I will finish with this picture, which I think is a great symbol of what we try to do in eTranSafe. And with that, I think I'll stop here.